Hey everyone, today we're going to talk about SDS Page. What is SDS Page? It's a biochemical technique that we use to separate proteins based on molecular weight or to find subunits. A great example is hemoglobin as a tetramer, meaning it has 4 subunits. So we use electrophoresis, and that's going to tell us the first part to understanding what it stands for. Page, gel electrophoresis. So you have some type of gel, and on that gel, you have charged particles, or proteins we're talking about, in an electric field. And that electric field is going to draw the proteins from negative to the positive end. Smaller proteins in this gel will travel faster through the matrix. That's the gel, right? And you have other factors such as density and shape. But the best thing about SDS Page, it uses sodium dodecyl sulfate, which reduces the influence of structure and charge, leaving the determinant factor to polypeptide chain length. That is exactly why density and shape don't really matter, because we have SDS in there. And why does that not matter? Because you got to understand 2 concepts. If you have this protein, this poorly drawn protein, has positive and negative charge all over, and in between the groups are disulfide bonds. We use 2 ingredients. We use the beta-mercaptoethanol, and we break these disulfide bonds. We add SDS, and it denatures the protein, getting rid of the shape. It then becomes a rod shape, and the SDS, often referred to as an anionic detergent, attaches to this rod-shaped protein and gives it an overall negative charge. That's perfect because it allows our gel electrophoresis, like I said, to flow from negative to positive. Approximately all proteins at this point have the same charge, density, and shape. Now I refer to what a cartoon illustration of a gel looks like. So think of a square, and the gel is made of polyacrylamide. We'll go into a little bit further, but at the top there are wells. And in these wells, it's dyed with methylene blue that gives you this blue color. And you have a pipette, and you'll pipette your protein with the methylene blue with everything else into these gels, or wells. And in the gels, you have 2 layers, the stacking and the separating gel at different pH. It's important. In a second, I'll tell you. But this is called a discontinuous system, named after the famous scientist. Now, you have an electrode buffer. So imagine in this gel, you have something surrounding it, allowing things to flow. This electric buffer at 8.0 pH. Now that's important. Keep in mind the pH levels. So we go on to, what is a stacking gel? The stacking gel prevents protein samples from diffusing into the electrode buffer. This is because the stacking gel is at a lower pH, which favors the protein, allowing the protein to flow from the higher to the lower pH. This is great because imagine you have proteins of different sizes and you have different things in there. They're going to flow at different speeds. So if you have a protein at the top, you don't want it to start all the way at the top. You want it to start at the same position. The lower pH allows it to go to the bottom of the well. Then you have what I referred to was the electric buffer, but it's the migration buffer. And it contains buffers like tris and glycine. Glycine responsible for viscosity, and tris gives you the chloride ions. Glycine is important because it's a Zwitter ion, which means depending on the pH, it can be neutral, having both positive and negative charges, or when it's high pH, it has a negative charge, an overall negative charge. So why stacking? Why was it important for me to mention all this? Well, when you look inside the stacking gel, you're going to find that chloride goes down first. The chlorine atoms go down. And the protein complexes comes thereafter and followed by glycine. And it creates a stacking. So Cl and then protein on top and then glycine on top. Stacking. Then when you enter the separating, you're going to see that the glycine switches position in the stacking to be in front of the protein complex. And that's why it's important to note that it was a Zwitter ion. Because at a higher pH, the pH of the separating gel being higher than the stacking gel allows the Zwitter ion to have an overall negative charge flowing faster to the bottom because of the positive charge at the bottom. So you run the gel, and the gel at the top of the wells will go down. And on the right side or the left side, depending whether you put it face down or face up, is the markers. And the markers will flow. And this will allow you to tell what in relationship between this and this is the molecular weight. For example, if each one was 100, so if there was a bar right next to this one, you could say that the molecular weight is 100 kilodaltons. If it was next to this one, it would be 200. So if I said this was 300, the next one's 400, this or all these are in between should be 300 to 400. Now note that this is not actually 300, 400. Why? Because methylene blue flows faster than all the proteins. The proteins have different size charges. So actually you can't see the protein in here. You don't know where it is. But this gives us a good reference point. Because if we can't see the methylene blue, that means, or it's possible, that the methylene blue has already flown off and we might lose some of our protein on this figure. So we want to see the methylene blue at the bottom. That means that most of the separation has occurred. So you move on to the last part. Well, what you would do is you would stain it with something called the blue staining. And this allows you to visualize the protein. So you're going to see this new stuff and everything turns blue. As you know, there's different colors here. That's because you stain it blue. And now you can see the actual protein. But what I didn't mention was about the acrylamide. So why is acrylamide important? Well, you have a few things. So when you make the gel, you can have a precast gel, which is something that's pre-made. But often or not, you have to make your own gel. You have acrylamide and bisacrylamide. Well, Kevin, they're kind of the same thing, aren't they? They're both acrylamide. Well, kind of. When you put acrylamide and bisacrylamide, the ratio matters because if you have something with more pores or something with increasing size with more cross-linking, you're going to decrease the pore size, and that means that basically it's going to be harder for the proteins to go through. So you're going to basically do this so if you have a fast moving protein, small molecular weight, you want it to be thick. If you don't have something that's fast, you're going to have it thin if you want it to flow a little quicker. Now that's relative, but something to consider, the ratio. Cross-linking means the joining with covalent bonds. And the reaction responsible is the vinyl addition polymerization. Now, it's initiated by a free radical generation. And why is that important? Because acrylamide and bisacrylamide is not just going to sit there and have cross-linking. It needs something. So we use APS, and APS is the initiator of that free radical generation or that polymerization, also known as ammonium persulfate. So you're sitting there, you put it in, and it takes a while, and you're wondering why it takes a while. So we do something called TEDMED, and you usually add this last. TEDMED, tetramethylethylene diamine, which is increasing and accelerating the rate of formation of free radicals from the APS to catalyze the polymerization. So as you add TEDMED, if you increase the amount of TEDMED, it's going to increase the rate. That means that if you have it in a beaker and you're making a gel so you can pour it into the casting tray, which is kind of like to get the shape right, if you put a lot of TEDMED, it's going to solidify in there because you're increasing the rate. But if you put a little, it's going to slowly form. You don't have time, understand how much TEDMED you need to add. Now, TEDMED is subject to oxidation, which means certain factors such as reactivity you will lose over time. So that's most of what you need to know for SDS Page. If you enjoyed the video, like below, or if you had something you found particularly wrong, leave a comment below and I'll put an annotation up. But thank you for sticking around, and I hope you enjoy your time making gels with SDS Page.